Good day, everybody, and welcome to the MassBio Innovation Week. We have an antibiotic day at the MassBio Innovation Week. How great is that? I'm Martin Heidecker. I'm Chief Investment Officer at the AMR Action Fund. I'm also a board member at MassBio, just for disclosure. You can imagine how excited I am to be able to speak today in this keynote speech about our role in helping to fight bacterial diseases and infections. The title of my presentation is Redefining Massachusetts' Essential Role in the Global Fight Against AMR. I would like to start the presentation with a little bit excursion into the history of antibiotic discovery. They were pioneers in the field who started to research and who were interested in finding out what causes diseases. Theodor Escherich, for example, is known as being maybe the first pediatrician. He was very interested in finding out what makes children specifically sick. And to honor his work, bacteria was named after him, Escherichia coli. I don't know whether you have ever wondered where this strange name comes from. Well, it's obviously named after this scientist who was born in the beginning of the 19th century, started his work with Rudolf Virchow, and then went to Vienna to do his work. Other bacteria are also named after great scientists. Klebsiella pneumonia, for example, named after Edwin Klebs. Albert Neisser gave his name to another bacteria, Neisseria gonorrhoea, a very difficult to treat pathogen that today is resistant against many molecules we use to treat infections. The only person on this slide who's still alive is Paul Bohr-Baumann. After him and his wife, Linda, I couldn't find a picture of Linda in the World Wide Web, so it's not meant to be exclusive here. But Paul and Linda Baumann gave their name to Acinetobacter baumannii, a pathogen that's causing a lot of problems, specifically in infections of the lung. And if this bacteria becomes carbapenem resistant, it really is a huge issue today to treat patients suffering from Acinetobacter infections today. Many of these bacteria are members of what we call the escape family. These are the very difficult to treat, very difficult to kill pathogens that are responsible for millions of deaths worldwide. So you can imagine the discovery that bacteria are responsible for diseases triggered a lot of research aiming towards finding solutions for those infections. It all started in the 40s when Alexander Fleming, who you all know, discovered that penicillin, a natural substance secreted from a fungus, can actually kill bacteria. And we still today use penicillin to treat infections. This discovery triggered what we call the golden age of antibiotic development in the 50s, in the 60s and 70s. Many drugs were developed that we still use today. And everybody knows that antibiotic discovery and research saved so many lives, and it's estimated that roughly 20 years of our lifespan we thank the antibiotic development. It's very interesting, whenever I talk to people, and I invested in other areas before, antibiotics, everybody has used antibiotics. So it's very unusual that you talk to an audience and everybody knows what you're talking about, different from cancer and other diseases where a minority of people suffer from. Antibiotic infections, everybody of us had a Staphylococcus aureus infection already, and we used antibiotics, and that's why we are still around. What you see on this slide and on the bottom of this slide, that soon after antibiotic molecules are introduced, we see resistances. We see bacteria who cope with the threat, who deal with the molecule. They change their proteins and they become resistant to the molecule that was developed. When penicillin was introduced in the 40s, already it was known that some bacteria cannot be killed by this molecule. And when you look at the vancomycin, an important antibiotic in the 70s, in 88, there were first resistances reported. Today, we have resistances against nearly all drugs that we use, and it becomes more and more difficult for many infectious disease specialists to treat their patients suffering from multi-drug resistant pathogens all over the world. This is a problem in the United States, it's a problem in Europe, and it's a problem in low and medium income countries. So a global threat. And what are we doing about it? The history of antibiotic development has, also a long tradition in Massachusetts. I don't know whether you are aware of the Coconut Grove nightclub disaster, and I learned about that when I was looking for some interesting information for this presentation. In 42, obviously, a big fire started in a nightclub in Boston, killing more than 400 people. Many of the survivors suffered from severe burns, and penicillin was first time in a large scale used to treat civilians by physicians in Boston. Penicillin by that time was used already by the military. The army used it to treat wounds that soldiers suffered, but civilians first time on a large scale experienced this treatment in the 40s in Boston. 68, I think another landmark study in the New England Journal of Medicine described an outbreak of a multi-resistant pathogen, methicillin-resistant Staphylococcus aureus, that still today causes a lot of problems for us in hospitals, first time described by researchers in Boston. Many of us know Dr. Stuart Levy at Tufts, and he basically showed in a groundbreaking work that, well, resistance can jump from livestock to humans. And this is obviously a threat that we all exposed. And still today, when you prepare a chicken, you better clean up your kitchen after that, because you might get infected with difficult to treat Salmonella bacteria. Cubist in the 90s, and this is my last landmark here, launched in Lexington and obviously contributed significant to development of antibiotics, launching a drug, Cubicin, and then being actually bought by Merck, which I believe is the last big exit that we have seen in the antibiotic space. So Massachusetts was always contributing to the history of antibiotic development. And it also contributed, of course, to the launch of startup companies in this space. What's the problem today? Let's spend maybe some minutes talking about what the issue is today. And I think that's very nicely described by this one slide, which actually compares the investment in oncology here in blue with the investment in antibacterial companies. And you can clearly see that there's a huge difference, and the gap is getting broader. Nearly $7 billion were invested in 2020 into companies developing oncology drugs. 200 million were given to companies developing antibiotics, and this low number is very steady over the last decade. In a time, remember, where we face all the development of resistances, we do not invest enough money to stop this problem. This is clearly seen when you look in the pipeline, right? No money going in, well, not so much can come out. And when you look at the 2020 figure here, you can see that there's only one NCE with a non-novel target that started a clinical trial in an antibacterial indication. and there's 2 NCEs with novel targets, but it's only 3 compounds that entered into the clinic in 2020. Clearly a reflection of the lack of funding and research that we see in the area. Even, and that's maybe even more shocking, even if we get drugs approved, it doesn't mean that they're necessarily launched. And what this graph here on the right side shows you, the approval and commercial launch of molecules per country, and the data ranges from 2010 to 2020. So in the United States, only 17 of those molecules have been approved and launched. But when you look into Europe, you can see that in some countries, this number is even significantly lower. Germany 7, Italy 7, France 8. What does it mean? Well, it means that even if you get approval for a new molecule, even if you get through the clinical trials, it's not worth launching a drug, which actually indicates very clearly how broken this market is. If you cannot make money by launching a drug, well, you don't do it. And this is reflected in this slide. So I think we need changes in the economy, and we need changes in reimbursement when it comes to antibacterial development and antibacterial drugs. So this is the situation. I think that's the reason why we are here. This is our team. We are the AMR Action Fund, and we just started roughly 1.5 years ago. We raised $1 billion from LPs, mainly pharmaceutical companies, but also some non-profit organizations. And we want to invest this $1 billion in companies in Europe, in the United States, also in Asia, companies that develop drugs against bacterial infections. Those infections should be critical or high priority according to WHO and CDC lists. So it should be problematic bacteria, and some of them I showed you at the beginning. And this makes us actually the world's largest private-public partnership, which is pretty exciting, and we're very proud of that. We have offices in Boston and Basel, and we still continue to expand our team. Our goal is to invest in 2 to 4 novel companies a year. When you look at what is our mission here? Well, our mission is to really enable the launch of novel antibiotics. You saw the thin pipeline, you saw the lack of investments. This is where we would like to focus on. Innovation in this space, in the antibacterial space, is driven by small companies. It's driven still by startup companies. Many big pharma companies stopped research in the field. That's why we are an investment fund. We invest in equity in this company, enabling them to get data and to launch the drugs that we so desperately need. Our goal is achieved, our mission is achieved, if we enable the launch of 2 to 4 novel antibiotics, which doesn't sound a lot, but it actually would make a big difference for all the patients suffering from those multi-drug resistant pathogens today. I mentioned already we have headquarters in Basel and in Boston. And what are we doing in maybe a little bit more detail? It's shown on this slide here. I think I talked about most of it already. We invest in biotechs. We want to enable the launch of drugs. We also talk about the reforms that are needed. And remember the slides, you know, even not being worth a launch, if you have developed the drug, that's an issue we have to fix. I think we need market reforms. We try to talk with stakeholders making them aware of the problem and encouraging them to come up with solutions for this issue. What we have done in the past, we started in 2020, we have 2022 now. So we have invested in 2 companies already, and I think it gives you a good idea of what we actually like to do. One of those company is a more innovative company. They use phage, the natural viral predator of bacteria, and Adaptive Phage has actually collected a number of phages, has characterized them, and put them into what they call a phage bank. And they just started with our investment. They started clinical trials in chronic indications, diabetic foot osteomyelitis, and prosthetic joint infections. Those patients suffer sometimes from infections for more than a decade. You get a hip replacement, you get a knee replacement, you get an infection, and you cannot treat the pathogen anymore. You will end up in today's situation with surgery. You will lose your limb. Adaptive Phage is recruiting those patients, is infusing their phage therapy, hoping to have an impact on the treatment of those difficult to treat pathogens in the joint of those patients. The other investment we made at around the same time last year, Venatorix, a more classical small molecule company. They developed what we call a beta-lactam, beta-lactamase inhibitor combination. They just got phase 3 data in a CUTI trial, superiority data. And with the money that we invested, the company is preparing for the launch of this drug. They just have received a BARDA grant. I think those grants, BioShield award, those grants are very important to support the development of those molecules, because it's so hard to find investors for those indications. So I think this gives you quite a nice overview of what our interest is. We hope to be able to announce a couple more investments this year. Let's see how this works out. We are already at the beginning of December, but I'm very optimistic that soon there will be more than 2 portfolio companies in our portfolio. Well, why Boston? What's Massachusetts' contribution to this? This is the Mass Bio Innovation Day. Well, I think Massachusetts has a lot of great ingredients that you need to actually solve this problem. Look at the AMR community from a small business organization point of view. And it's just, you know, a snapshot here. It's not meant to be all-inclusive. It just, depicted some examples here. Spero Therapeutics, publicly listed company. We have some early companies like Arietis Pharma, Vedante Microbiome, as well as Ceres, focusing on C. diff infections, also difficult to treat in many circumstances. Diagnostic companies are here. I depicted 2, and others. So based on the strong academic background that we have, and I didn't want to put like MIT and Harvard on this slide because it's obvious, but we have other very great research institutions in the space. We have Tufts, we have Boston University with a long tradition of antibiotic research. I think a lot of great, scientific insights that you need to actually build companies and find solutions for the problem we are facing. I also wanted to talk a little bit about deal making, because I think we need deal making to create exits. As an investor, as a venture fund, well, I need the exit. And it looks like the exits in this space, and I think we can talk very lengthy about this, it changed over the last decade. When I came to Boston 10 years ago, it was like Merck buying Cubist, $8 billion on the table, big deal. We don't see those anymore. And today, I think there's still deal-making activity, and I showed some examples here. Again, This is not holistic, but it's some examples. Tetraphase was acquired by La Jolla in 2020. Entasis was acquired by the main shareholders Innoviva, and then launched, merged with La Jolla. I didn't show that here. GSK did a deal with Spero, investing, but also licensing deal. And again, GSK acquired Affinivax. So we see deals. The volume of this deal is far away from the 8 billions we saw previously. I hope that the enthusiasm of the community and the pharma buyers will come back for this space one day. When you look at the investor community at the end, we have the small biotech, we have the scientific, the academia background. Do we have investors in Boston and Massachusetts? Well, I think we have. First of all, we have the AMR Action Fund. As I said, we're headquartered in Boston. We have $1 billion under management that we want to deploy into antibiotic companies. But we also have non-profit organizations, CARB-X, which many of you are familiar with, and Kevin Utterson, since years and decades, is very active in this space, investing non-dilutively in earlier antibiotic opportunities. Many of the companies we look at actually have CARB-X funding at some time, and also one of our portfolio companies, Venatorix, received significant funding from CARB-X. The Repair Impact Fund, also in Boston, and Alex Engels, I think, is also very well known to many of you, investing in earlier stage antibiotic companies. And as you can imagine, we very often talk with Alex and also with Kevin and Erin about investments, about what we need to see in companies in science to be able to invest and to enable the launch of novel antibiotics. So around this, only 3 companies, we actually have roughly $1.5 billion that can be invested in antibiotic company. And we also cover the full spectrum of development from very early to very late. The AMR Action Fund, as you might know, is focusing on clinical stage opportunities only. And of course, we collaborate close, and I think we need to have this alignment on the policy priorities. Everybody in this space is aware of, we need those changes that make this market attractive again to succeed in our desire, our motivation to launch novel drugs. And those launches, by the way, cannot only happen in the United States. We need to make sure that those launches are happening worldwide, because pathogens can jump, as we have seen in a long time ago, from chicken to human, but they also can travel from India to the United States and from Europe to the United States. Bugs don't stop at borders. They travel with us. This is a little bit what I wanted to share today with you at this Antibiotic Day. So really the invitation from our side, we would like to contribute to the development, to the launch of novel antibiotics. We are aware of our focus is later stage, but we're very open to engage in discussions with earlier stage companies, even with academias, with non-profit organizations, the venture community, whoever wants to reach out to us. We're really happy to talk with you. We have one goal. We need to fix this problem. We have 1.27 million deaths today, deaths caused by multi-drug resistant pathogens. We will have many, many more in the near future if we don't get the pipeline filled. If we don't fix this broken market, please reach out to us. This is our website. This is my email, and we're looking very much forward to having interactions with you today at this Antibiotic Day, but also beyond. We're around. Please reach out to us. Thank you very much for your attention.
Hi, everyone. My name is Gauri Nair, and I'm the Vice President of Innovation at MassBio. Welcome, and we are happy to have you here today. Thank you for giving us the opportunity to connect with you on this very important Infectious Disease Day of MassBio Innovation Week. These were two wonderful talks by Martin Heidecker from the AMR Action Fund and Aaron Duffy from CARB-X. Thank you to AMR Action Fund, CARB-X, and Repair Impact Fund for the sponsorship of Infectious Disease Day. I encourage all of you to go to the sponsor booth for more information about everything they do. There's lots of information available, and hope you'll have a good visit to that booth. Before we get started with the presentations, I have a few housekeeping items. All attendees are muted. The presentations for each company will be for 20 minutes, and there'll be a 5-minute Q&A after that. If you want to ask any questions from the presenters, please put them in the chat function, and we'll be sure to ask them once the presentations are completed. Thank you very much, and I hope you enjoy the next presentation. It is my pleasure to announce Sat Shukla from Spero Therapeutics, who will be presenting on their platform and the company. Thank you.
Fantastic. Thank you, Gauri, and thank you, MassBio, for having us and hosting us today. It's a pleasure to be here. Let's move into past our usual forward-looking statements. So, Spero Therapeutics, the company is approaching a decade in the space. We have been a public company for approximately 5 years now. We are focused on targeting areas with high unmet need and strong commercial prospects. So we try to develop medicines that possess and communicate both therapeutic and economic benefits. Our pipeline addresses areas where there are currently few or no approved alternatives, and we are always looking for ways to continue to strengthen that pipeline opportunistically. Our lead asset is SPR720, potentially the first novel oral first-line therapy for NTM, non-tuberculous mycobacteria disease. It's a oral therapy that targets DNA replication, and we have been very excited to announce the initiation of a phase 2 for this therapy recently. It's a proof of concept study. Clinical site has been opened, and we expect to screen and treat patients in this quarter. This study follows phase 1 SAD/MAD study and in vivo efficacy data that support, we believe, a differentiated profile. In addition to our lead asset, we also have other assets that have been fortunate enough to bring in partnerships with some leading entities in the industry. Tebipenem HBr is our oral carbapenem, potentially the first therapy for patients resistant to current oral therapies. And this is an asset that, as Martin mentioned earlier during the MR presentation, recently entered into a global partnership with GSK that supports its clinical development through approval. Our other asset is SPR206, which is a next-generation polymyxin. This is a therapy we believe will be efficacious against resistant bacteria, resistant pathogens being another topic we have covered a bit today, and I suspect we'll cover later as well, but without causing the nephrotoxicity that is currently associated with many therapies in the space. This asset currently has partnerships outside the United States with Pfizer, who invested $40 million into Spero last year, along with a license for this asset, Everest Medicines. And that partnership funding, along with government funding, provides support fully for phase 2 of this asset. During this time, Spero retains the US rights. So this slide provides just a visual representation of how we see our pipeline. It's a late-stage pipeline addressing high unmet need areas. with the de-risking common to pipeline that is later stage. SPR 720, as I discussed, is now initiating phase 2. Tebipenem HBr expects to be in phase 3 in partnership with GSK next year. And SPR 206 expects to be in phase 2 by the fourth quarter of 2023. On the right side of the slide, we do list some of the parties we have been fortunate enough to collaborate with in progressing these medicines. So these assets have undergone scientific scrutiny from a variety of government entities, BARDA, NIAID amongst them. And on the commercial side, GSK, Pfizer, who have also, in addition to the scientific underpinnings of these assets, have also looked at the commercial opportunity, potential commercial opportunity for these assets. In triangulating something for what we want to progress our assets, again, starting with that high economic need and confluence with strong economic benefits. Some of the assets listed here in between are other launches that have had success in working in that confluence. Those are launches that we hope to replicate through progressing the medicines that we are bringing to market. And we also generally focus on therapies in an outpatient setting. So bypassing some of the challenges and focusing on non-DRG reimbursement as opposed to hospital-based reimbursements. So that's the overall portfolio. Moving forward to our lead asset, SPR 720. So the chart on the left shows the cycle for this disease. NTM is a chronic infection. People inhale it. Folks who took showers today inhaled it probably from a shower washer. But for people who get infected and whose systems lead to symptoms and then a greater passage for this disease, it can have debilitating pulmonary symptoms. So on this chart, the only approved therapy currently is Insmed's inhaled Arikayce. That's on the refractory side of the chart. On the earlier side of the patient journey, patients do need therapies, and currently there are no approved therapies in that space. So SPR 720 has orphan drug designation, and if approved, could be the only novel first-line oral treatment for NTM. Looking at the landscape, so NTM in the United States is approximately 95,000 patients. Globally in major geographies, that number approaches 250,000 patients. And 75% of these are in the non-refractory space. So again, an area with no approved therapies at this time. SPR 720 has potency against a range of these NTM pathogens. And once daily dose is supported by clinical and non-clinical studies. So selected 500 megs to 1,000 megs once daily dose range for phase 2 is supported by in vivo and in vitro PK/PD models. And additional BAL study data in non-human primates supports lung exposure. Macrophage data has shown intracellular and extracellular activity. And these are accompanied by supportive safety and tolerability data that have led to our design for phase 2 at this time. Let's talk about potency first. So this chart shows a concentration of SPR 719, the parent compound to SPR 720, in relation to amikacin for activity against a variety of NTM species. The metric here is minimum inhibitory concentrations. The degree of drug required to reduce infection by roughly 90%. So a smaller number is better. And you can see on this chart that SPR 719, the degree of drug compared to amikacin is substantially lower, indicating higher potency potentially for this drug. That is supported with additional data as well in a murine chronic infection model. So showing this chart, this shows bacterial burden on lungs on a log scale. And since it's a log scale, the reduction from therapy is not proportional. It is in fact exponential. If you look at the untreated patients on this chart, SPR720 monotherapy showed a reduction exponentially on lung burden from pathogens in this study. And in combination with the clarithromycin and ethambutol, the reduction was even more substantial. So that led us to conclude that this therapy potentially has strong potency, either as a monotherapy or in combination with other agents, and could substantially change the landscape of treatment for this disease. That data, the potency data, was combined with roughly 100 patients dosed for tolerability studies. So SAD and MAD studies showed no serious adverse events, no clinically significant lab findings. Side effects, if they occurred, were very minor and usually on the GI side, very common with these types of therapies. And only one discontinuation at very high dosages at 1500 megs, which was asymptomatic, monitorable, and reversible. Looking at the landscape again. So SPR720, the target would be first-line patients. That's the treatment-naive and treatment-inexperienced patients. No current approved therapies. And patients here often delay treatment because the regimens of antibiotics are generally poorly tolerated. So those delayed treatments are due to intolerability, inconvenience, and ineffectiveness. By the time that disease progresses to refractory, where again there's currently a single approved therapy today with inhaled Arikayce, the treatment benefits are still limited because of declining lung function, additional comorbidities, and also the AE profile for this therapy is not gentle. It's tough on patients. That leads us into the design for our phase 2 proof of concept study. This is a study where we expect to treat treatment-naive and treatment-inexperienced patients with NTM due to MAM. Approximately 35 patients, 4 treatment arms, 3 of those randomized and blinded, placebo, 500 milligrams, 1,000 milligrams. And then for additional PK data, 4 patients in an open label study. The objective of this study will be to continue evaluation of safety and tolerability and building on the data we have seen so far on plasma PK. But more importantly, the drug's impact on patients, microbiological response now, and how patients feel and function, which is a crucial component for this therapy in the space where even approved or other regimens simply are not conducive to how patients feel and function, even with those therapies. The study commenced with clinical site initiation this quarter, and we're very excited about that. We do expect, as I mentioned earlier, our first patient screen and dose in this quarter. And it has the potential to be the first to show early microbiological response as a standalone agent versus placebo, which would be, again, a great outcome as the study progresses. That's a summary of our lead asset. Moving on to our partnered programs. Oral carbapenem, tebipenem HBr. This is the asset that we recently announced a partnership with for GSK. Again, significant opportunity both from a therapeutic and economic side. from a therapy side, the lack of an effective oral treatment in complicated urinary tract infections leads to a heavy burden on patients and the healthcare system. There are more ER visits, unnecessary and unwanted hospitalizations, more hospital days. And if approved, a oral carbapenem in treating roughly 2.7 million of the 8 million patients who suffer from urinary tract infections annually could be a substantial game changer. Tebipenem HBr has QIDP exclusivity and IP protection through 2038. And now moving into a phase 3 next year gives us plenty of time to develop and then hopefully bring this therapy to market over the next few years. Results from the ADAPT-PO phase 3 for this study were recently published in the New England Journal of Medicine, which demonstrated that oral tebipenem was well tolerated and non-inferior to IV ertapenem, which was its competitor in adult patients with CUTI and acute pyelonephritis. And now going forward, in this collaboration with GSK, GSK has received an exclusive license to develop and commercialize tebipenem globally, with the exclusion of certain Asian countries, which are held by Meiji Seika, our partner. Spero is responsible for execution and costs for phase 3 in the United States, and then GSK will be responsible for execution and costs of commercial activities in the United States and outside, excluding the Asian countries mentioned earlier. This transaction closed on November 7th, and for financial terms to Spero, it brought in $75 million in initial funding, partly as an upfront milestone, and an equity investment in the company, leading to GSK becoming substantial shareholders in Spero. And going forward, up to $525 million in development, commercial, and sales milestones. 150 million of those milestones are development milestones that we expect to bring in upon initiation or completion of a set of activities in phase 3. So as that study starts next year and proceeds over the subsequent months, substantial portions of this funding will come in to support that spend. And then $150 million upon first sale if the therapy is approved on a global basis. Beyond that, if the therapy is approved and then goes through growth, Spero will receive up to $225 million in sales-related milestones upon completion of annual sales milestones. So if this therapy reaches $100 million in sales, Spero gets part of that $225 million. Similarly for $200 million, and so on and so forth. The company would also receive royalties, and on higher sales levels, these royalties reach low double digits and phase out the milestones, so bringing in economics to Spero from this asset. Moving on to our other asset, the direct acting IV potentiator, SPR206. SPR206, also has the potential to address a very high degree of unmet need in patients. Carbapenem resistance is something we heard a bit about earlier. Again, I suspect we'll hear more about it later as well. But obviously an area where there is significantly higher mortality, and the current regimen of treatment, carbapenems, polymyxins, and aminoglycosides, aminoglycosides and polymyxins, of course, cause nephrotoxicity in many instances. So for resistant, carbapenem-resistant pathogens, SPR206 has the potential to be not just an efficacious therapy, but also a well-tolerated therapy. Preclinical data are very promising for efficacy for SPR206 against a variety of pathogens. Here, these charts show SPR206 against meropenem and colistin in a variety of pathogens, and the activity is clearly strong. This is in congruence with the phase 1 SAD-MAD data on nephrotoxicity and about study data that showed lung exposure. So again, the drug is getting where it needs to get to without causing many of the AEs that historically have been a bane on these types of therapies. Going forward, these preclinical studies, the SAD-MAD trials, the BAL study, and the renal impairment study now have led us to develop a phase 2 for this asset, starting in Q4 2023 as expected. And to reiterate, this phase 2 is fully funded by the partnership support from some of the great partners we have been able to bring in to collaborate with us at Spero. So on the corporate overview, I'll keep this short, other than to note that we have showed up the balance sheet to make sure that we are able to progress this pipeline, this late-stage pipeline through exciting developments over the next couple of years. We announced a $50 million company cash balance at the end of 3Q this year. But that's before the $75 million from the GSK upfront and equity investment came in. That investment, along with the anticipated development milestones for tebipenem from GSK and non-dilutive funding from our other partners, gives us confidence that we have cash flow sufficient to fund operations and CapEx beyond 2024. So at the end of the day, the balance sheet for any biotech like ours, its biggest role is to make sure that medicines are getting to people. And with tebipenem going into phase 3 next year, SPR720 already initiated its phase 2 with a data readout in early 2024, and with SPR206 entering its phase 2 next year. We are delighted to see many clinical developments and potential outcomes for these assets during our current cash runway. Very briefly, this just lists our management team. My colleagues have been instrumental in bringing several therapies to market in our space, and we are very proud of that. We look forward to building on that with our pipeline at Spero. And I'll also put a plug in that these colleagues, along with our other colleagues at Spero, are terrific people, and it's a pleasure to work with them. That's really the end of my prepared remarks, so I'll be happy to take questions. Thank you very much for your time.
Thanks, Saad. That was a wonderful presentation. Fascinating, absolutely. What a great journey.
Thank you.
So I will be asking questions as the moderator. The first question is, what advice does Spero Therapeutics have for companies that want to play in the infectious disease sandbox?
No, thanks for asking. This may sound biased given the presentation I just provided, but I think that confluence of a strong therapeutic need while making the case that there is a real economic market for these therapies is something that I think we can never say too often. In our AMR presentation a little bit earlier, we saw a chart comparing oncology assets to the ID space. And prior to Spero, I was the CFO at an oncology company. So certainly the difference in perception has been very clear to me. But I think, we go back to highlighting that this is an area of great and critical need, and if executed appropriately, can bring in significant economic benefits to the companies bringing these medicines to market as well, and to obviously their investors. So certainly the $8 billion transactions have been few and far between recently, but it is great to see on the investor side focus turning again to this area, and that's just something that we, along with other new potential parties in the space, should continue to build on.
Fantastic. And what's next for Spero in terms of collaborations or, deal making?
Oh, well, again, thanks for asking. We were very fortunate to bring in the partners we have brought to the table so far. I think at the end of the day, our mission is to bring these medicines to patients, whatever form that takes. Currently, for SPR206, we retain US rights, and that's something where we will continue to look for opportunities internally or externally to move that program along. SPR720, currently we have global rights, so we will see what we can get done. Again, it's a disease with 250,000 patients worldwide, huge incidence in Japan. It's something that right now is, as data percolate out in that phase 2 study, it will give us more shots on goal on bringing that therapy to market. And I think, you probably noticed on my very lead-in slide, we have opportunistically built our portfolio and pipeline over time. We are delighted to have this late-stage pipeline today, but of course, we are going to continue to strengthen it in adjacent spaces or whatever meets that confluence of therapeutic and economic benefit. So now the company is well-funded, the balance sheet is strong. And so either on out-licensing, in-licensing, or other venues, we are excited about the prospects for this going forward. And so we hope that 2023 and 2024 will be great years, either for execution or for BD for us.
Yeah. The goal of Infectious Disease Day at Innovation Week was really to increase awareness about AMR and what can be done in this space. What else do you think that the ecosystem and organizations such as MassBio can do to help?
Well, I think reiterating the messages that we are hearing today are really important. I think over the last few years, there has been a tendency for uninformed parties to walk away from the space and these therapeutic areas. And so reminding people of the tremendous science, the tremendous people who work in these areas, and the potential benefits, both from an economic stage and from a public health perspective, I think that's a function that places like MassBio can continue to serve, and really, no other types of those entities can. So that is something that we are excited to collaborate with you going forward, and I hope others are there as well.
Thank you. Well, thank you, Sat. And we'll now move on to our next presentation, and we look forward to a continued relationship with Spero.
It's been a pleasure. Thank you, Gauri, and to our colleagues at MassBio.
With that, I want to introduce our next presenter who will be joining us today, Lloyd Payne, President and CEO of Arapath. Lloyd has flown in all the way from UK, and I'm very glad to host you here. Thank you for joining us today, Lloyd.
Okay, thanks very much, Gauri. It's a pleasure to be here. I hope you can hear me well. I'm just going to switch to presenting mode on here, so I hope you can see my screen. And it's great to be here. So as Gauri said, I'm the president and CEO of Arapath, Lloyd Payne. And I'd like to start off by thanking the organisers for giving me the opportunity to introduce you to Arapath today. So we're very excited to be able to do that. So what is Arapath all about? We're a Princeton University spin-out company. Our primary and core focus is delivering new classes of anti-infectives to the patient, and more specifically addressing the urgent challenge of antimicrobial resistance that we've already heard about today. We announced the closing of our seed round of $20 million back in March earlier this year, and we're delighted and privileged to be supported by a fantastic group of investors and co-led by Insight Partners, Boehringer Ingelheim, and Innospark, and also bringing together Viva BioInnovator, PTX Capital, Northeaster Ventures, and Arimed Capital. So I'd just like to spend a few moments really talking about the global health challenge of antimicrobial resistance, which is indeed very significant, and the challenge, the opportunity, and Arapath's response to that challenge. So as we know, treatment options are running out. There is an urgent need for new classes of antibiotics. And this has been covered really well in a number of reports over in recent times. And the WHO has done a really nice job of actually analysing the preclinical and clinical stage pipeline of antibacterials. And they've shown that over 80% of candidates that are coming through that pipeline are from existing classes of drugs where resistance mechanisms are very, very well established. And in those situations, you would expect to see rapid emergence of resistance. A second report that was very important that was published in The Lancet, called the Graham Report, analysed the global burden of antimicrobial resistance and reported actually that deaths attributed to AMR in 2019 surpassed 1.2 million, and also vastly surpassed diseases such as HIV/AIDS, breast cancer and malaria. So this is a really very significant problem. It's a problem that is growing and is often referred to as the silent pandemic. And this problem is getting worse, as I mentioned, and the WHO actually updated their analysis of the pipeline, and they've suggested that it's insufficient to tackle this increasing problem of emergence and spread of antimicrobial resistance. So where Arapath sits with this, and really where we see the opportunity and what we're doing, is delivering new classes of antibiotics, and very, very importantly, that represent under and unexploited mechanisms of action. So we believe to be differentiated here is important when we think about overcoming that cross-resistance issue. We're building a pipeline of differentiated antibacterial assets. We're focusing on significant unmet need, and I think that was really nicely covered by the previous speaker. And we cover that unmet need in terms of acute infections resulting from multi-drug resistant gram-negative bacteria, but also specific chronic infections. And this is how we look at building our early stage pipeline. How we do this, we have a platform technology, and we really harness the power of microscopy and imaging and couple that with machine learning from very early stage drug discovery through optimization and then onwards to candidate delivery. And I'll try and bring that to life in the next few slides and throughout the course of this presentation. I think the important point to mention here is we really believe that our platform holds significant potential in the discovery of new medicines to overcome drug resistance, not only when we think about bacteria, but when we think beyond bacteria also. As we all know, a great platform is nothing without a great team, and I'm privileged to be working with a great team. And here are some of the faces of the team here on this slide. We have a really experienced team with deep expertise gained from many years in pharma, biotech, and synthetic biology companies, with a real depth and breadth in anti-infective drug discovery, microscopy, computer vision, data science, and machine learning. So it's great to be able to work with this group, and they really are a great team. In addition to our internal team, we've assembled a world-class advisory team. There are some faces on this slide that some of you will recognize. The first is Mike Dudley. Mike is the chairman of our board of directors, and he's a fantastic guy to work with. He's also the CEO of QPEX, and Mike has really many years experience in antibacterial drug discovery, development, and commercialization. It's brilliant to have him on the team. Professor Zema Gitay is our founder, and he's a professor at Princeton University in quantitative microbiology. And we also have our SAB, Todd Black, spent many years at Merck as a microbiologist, and he's now at the TB Alliance. Barbara Engelhardt is a professor at Stanford University. And 2 new appointees to our SAB, Connor Coley, who is really a rising star in artificial intelligence and machine learning based at MIT, and also another well-known face as a chemist, Paul Hergenrother, well-known in the space, based at the University of Illinois. So I'm actually going to introduce a concept that we call domain-infused machine learning. And what we mean by that is really bringing all of this expertise and coupling that with artificial intelligence and machine learning. We don't see AI as a replacement to the human being. We really see the two working hand in hand and synergizing. So I don't need to tell this audience this, but as we know, discovering and developing new antibacterials is really hard. But probably more importantly, it's even harder if we do the same thing and take the same approach over and over again and expect a different outcome. So at Arapath, we look at things differently. We search differently, and we harness microscopy with machine learning, and we take a mechanism-first approach. Now, what do we mean by that? Coming back to the point I made earlier, we focused on new classes and clinically differentiated mechanisms of action. And that's really important. Traditionally, with phenotypic screening and trying to find new antibacterial starting points, in the past, the community has really taken an approach of phenotypic screening using a growth inhibition endpoint and screening enriched media. So really looking at antibacterial potency as the primary driver, and then counter screening against other important attributes. And the mechanism of action tends to be something that's looked at a lot later. Now at Arapath, we turn things on its head, and with our platform, it really enables us to investigate very deeply the biology at a very early stage and understand how compounds engage with their targets. And we do that alongside antibacterial potency and counter screening for other important attributes as well. And that's the real differentiator with Arapath, and I'll come on to that platform technology and how we approach that. So coming back to this point of domain-infused machine learning, we take an integrated data-driven approach to the discovery of new antibacterials and anti-infectives. And sat right at the center of this is our chemistry machine learning platform that we call ChemL. And this is really a way of us ingesting, curating, making sense of data, and using that data to drive decision-making through the drug discovery process from very early stage screening right the way through to lead optimization efforts and beyond. An important part of this is actually bringing into play publicly available data. And we ingest lots of data that's publicly available. And it's actually more difficult than it sounds, to be honest with you. So we spend a lot of time doing this, making sure that data is appropriate, cleaned and scraped, and then ingested into our pipeline. So we build predictive models, and we can apply those models of antibacterial activity, for example, in a screening sense. So for very early stage hit identification, which is actually very important when we think about finding new starting points for antibacterials, we can use predictive models to actually search chemical space, and this is very important. So I've put a little diagram here that was published in a paper by Heinz Moser and colleagues from Novartis, and you can't see this particularly well, but there's a green blob in that diagram which really shows the property space of typical corporate compound screening decks. And it's really shown that this property space doesn't overlap too well with what we might regard as antibacterial space that's really shown by the purple and blue dots in that diagram. So actually searching around in vast chemical space is really important to make sure we search in the right areas, but also perhaps search in areas that hasn't been explored before or has been underexplored. So we use predictive methodology to enable us to do that. So we select compounds, we screen those compounds, and we do that in a couple of different ways. We use growth inhibition screening against bacteria that are very important in terms of our target product profile. We're target product profile driven. We always think about the disease and that significant unmet need. Acinetobacter has been mentioned already today. But additionally, we screen against mutant and knockout strains because those data really help us understand liabilities of specific chemical features and structural features and property features that we either want to design out, or indeed if they're important to gram-negative uptake, we want to obviously retain them and design them in. In addition to the growth inhibition screening, and very importantly, we use microscopy and phenotypic screening to really get into the depth of the mechanistic details and how a compound engages with its target, and also to understand the potential novelty of that target at a very early stage. So that actually generates an awful lot of data. So we ingest those data into our ChemL platform, we refine our models, and then we can go back around and iterate, screen further compounds, and then ultimately identify compounds that we take into later stage drug discovery, into hit to lead and lead optimization. And again, our machine learning platform sits at the center of this and also helps us with in terms of informing decision making at these later stages. One thing I haven't gone into detail on, when obviously just looking at antibacterial activity is mono-dimensional, but we look at multi-dimensional parameter optimization. And cytotoxicity and ADME parameters are really, really important at this stage also. Cytotoxicity in particular, so when I mention that, I talk about mammalian cell cytotoxicity, because what we don't want to do is just develop a model that selects compounds that are broadly cytotoxic. So we always think about this in multi-dimensional space, and we use data to help us optimize and design our properties that are unwanted as well. So I'm just going to give one example of how ML models can help us select compounds for antibacterial activity in a screening sense. And this has been done before, but one thing I really wanted to point out that is a generalizable model is really critical, and models can be criticized for the fact that they just simply memorize compounds that look like the training set. Well, we don't want that. We want a model to actually work and essentially enable us to identify antibacterial compounds that are outside of the screening set, and the training set, sorry. And we ensure and validate that generalization by testing the model on compounds that look completely different to the scaffolds that are used to train it. And our model of gram-negative activity has been trained on over 1 million compounds, and we use that to predict the activity of new compounds that we wish to screen. So we're essentially prioritizing compounds for screening at a very early stage here. So if successful, such a model can meaningfully accelerate screening by returning higher hit rates for fewer compounds screened, and also provide the basis for an iterative screening approach. So the diagram on the right-hand side really shows you that we see enriched hit rates, and this is validation data, and we can see really good return rates for screening a smaller amount of a compound collection and selecting carefully, driven by a model, and actually identifying greater percentages of actives found. And we see that this is applicable not only just to a single strain of bacteria, we've applied this to several strains of bacteria as well. So coming back to the imaging platform, and this is a really important powerhouse of our platform. And just to explain this, we see this and regard this as something akin to going to the movies. And it's a bit macabre. What we do is undertake and watch bacterial autopsies. And we do this, we go to the movies and watch these autopsies in a high throughput multi-well format. So we look at thousands of cells per well and compute thousands of features for each cell when they're exposed to a compound. And what we can also do, rather than just looking at death as an endpoint, we can actually film very subtle morphological changes, i.e. how the cells are injured, and not just cell death. And that really speaks to the sensitivity of the platform, which we feel is very important. Again, this generates a huge amount of data, and we can rapidly reduce that resulting very complex data landscape and cluster the behavior. And this is really where machine learning comes in. we can use our data science platform to predict the mechanism of action where it's known, and or the novelty of the mechanism where it's not known. And we do that by comparing very rapidly against our known universe of hundreds of antibacterial compounds, which I'll come on to in a little bit more detail. So this proof of concept was actually published by our founder, Professor Zema Gitay, back in 2020, where he showed the discovery of a new dual mechanism class of compounds. But we've really taken this platform on and industrialized it and apply it with rigor to our drug discovery activities. So bringing this to life a little bit more, our imaging platform essentially answers four main questions. Firstly, does the new compound inhibit growth and or give rise to a phenotypic event? Secondly, does the new compound have a known mechanism of action or not? And if it does have a known mechanism, then what is it? What could it be? And what's predicted? And lastly, if it's not a known mechanism, is it novel when comparing to our proprietary training set? And I think the important point I want to point out here is that training set does cover vast mechanism space and also includes many compounds that are in the pipeline and also marketed drugs, and that's important. So on the right-hand side, the top picture, if you like, is our movie of a new hit. And what we can do very quickly is compare that movie with the movies that we've taken of known compounds and how the bacteria behave when exposed to those known compounds. And where the clever data science comes in is it tells us whether that movie has been seen before. If it has been seen, it will tell us what the mechanism of action potentially is. Or if the movie hasn't been seen, it will tell us that there's a high possibility that that mechanism of action is novel. And we have many, many movies in our reference library. And we do this quantitatively as well, so that's important to mention. So this is a busy slide, but it shows you how we look at mechanism of action space. We look at the cellular processes within bacteria, and our training set of drugs and antibacterial compounds covers a vast amount of this mechanism landscape, from really when we look at the highest level of cellular process and down the branches and to the leaves, and when we think about individual targets and groups of targets. And one thing I really want to get across here, that this imaging platform is really very powerful. We can classify mechanism of action where it's known at very high levels of accuracy of greater than 70%, not only at this high level, but as we go down these branches and to the leaves at the finest level of detail, retain these high levels of classification accuracy. However, and in the spirit of honesty and balance, as always, no one platform is perfect. We do see some challenges with weak phenotypes, which is perhaps not surprising, but the scope and the power of this platform is really wonderful when we use this in our screening mode, but also to assist in later stage drug discovery. So to bring this to life with some real data, and this is some data from, again, this is sort of early stage screening. So you can see the richness of data that we actually produce and the importance of the data. So we can identify compounds that have an antibacterial effect. We can classify and predict the mechanism of action where that mechanism might be known, but also we can predict potential novelty of that mechanism of action. So here are four examples. The four pictures are the movies that we refer to. And if we look at compound A in the table, the first number is an IC50 of antibacterial growth inhibition. That first number is actually against an E. coli cell wall compromised strain. The second number is against a cell wall intact E. coli, a wild type strain. So you may ask, why are we using a compromised strain? And that's actually quite important. So the reason being because it widens the net in terms of the ability to potentially find compounds that engage with important targets. But of course, we need to make sure that we have activity against wild-type organisms as well, and that's why we also screen against wild-type. And we don't just look at E. coli, by the way, we look at other bugs too. So compound A is particularly interesting because that movie has not been seen before by our system, and that's why we see a novelty score of 1. So 1 is the highest novelty score, 0 being the lowest. And of course, it doesn't have a predictive mechanism because it hasn't seen it before. So that compound is really interesting to us and something that we're investigating. Compound B is an example where we see activity in the compromised strain and also the wild type, but you'll see it has a low novelty score. So it's a high chance that movie has been seen before. And we do predict a mechanism, but we've not disclosed it in this presentation. But nonetheless, it's actually relatively interesting. Compound C is another example of activity in the compromised strain, but not in the wild type. But in this case, we actually see a modest novelty score, and we see a predictive mechanism really in terms of cell envelope organization. That gives us an idea of what's going on in that case. Compound E, finally, is a great example, actually, of where we don't see growth inhibition at the highest concentrations tested, but nonetheless, we see a phenotype. So it shows how sensitive this platform is. The novelty score is quite modest, but there's a suggestion of ribosomal aspects to that mechanism as well. So this is sort of gives you an idea of some examples of the type of richness of data that we see at a very early stage, and we use this to help us prioritize our activities in terms of hit expansion and follow-up. And in this particular table, compound A is actually really very interesting to us because of the novelty aspects that our platform returns. So I hope that's brought some of our platform to life and some of our early stage activities. And really just to finish up and talk about the opportunity, the outlook, investment and collaboration in the next 1 to 2.5 years horizon, and as we head towards our Series A in 2024. We are discovering and developing new classes of antimicrobials, again, really importantly with that emphasis on under and unexploited mechanisms, but really importantly coupled with clinical utility as well. Our target product profile focus is on significant unmet need, including multi-drug resistant gram-negative infections, acute infections, but also specific chronic infections as we develop our pipeline. Our pipeline and platform build is twofold. Obviously, we're moving compounds through, we're progressing our current compounds and enhancing that pipeline towards preclinical development candidate in the clinic. But additionally, a second and an important part of the value proposition within the company is building this technology platform, applying imaging and data science to new lead discovery and optimization, developing and applying a range of predictive tools for anti-infective drug discovery when we think about obviously antibacterials, but also we believe this can be applied beyond bacteria. And really just to finish up, areas for collaboration, we're very excited about the company and the progress we're making in this space. We're really very committed to discovering and developing new antibacterials, and we want to engage and build relationships with potential investors, collaborators and other stakeholders as we build the company, progress our programs and evolve our pipeline and platform as we prepare for that Series A in 2024. So I'll leave it there. I thank you for your attention today. I'll be really happy to answer any questions that anyone may have. Thank you very much.
Thank you, Lloyd. That was fascinating. Wonderful to hear about your platform to discover new antibiotics and anti-infectives. Quick question for you. What advice would you have for companies that are starting out in this space?
Yeah, it's a good question. I was listening to the previous speaker, who gave a really nice answer to that. And I would say, yeah, obviously this space is challenging. I think, you know, we're not naive to this, and I think we've heard a lot about the commercial issues here. But I think anyone starting out in this space, I really feel it's about ensuring there's a route to commercialization and development, but the focus on significant unmet need is critical. But also differentiation and how to approach this whole area. So And we feel if we bring this back to Arapath, the platform technology coupled with attaching that to this developing drugs that really are against that significant unmet need is really important. So anyone setting out in that space, I really feel that if you take that on board, I think you stand a fighting chance and really towards commercialization.
Well, that's fantastic. And I didn't see any questions coming in from the audience, any other questions, but and we're a little over time. There are a few comments of people wanting to reach out to you, and I'll be sure to communicate that to you offline.
Great, thank you very much.
Thank you so much, and all the best for your Series A. Looking forward to it, hearing more.
Thank you.
With that, I want to announce our next presenter, Milan Zdravkovic. I hope I pronounced your name all right.
Yes.
From Sniper Biome. He's calling in all the way from Copenhagen.
Yes, so let me see. I just want to share my screen. I think it should be projecting now. Is it working?
Yes.
Okay, very good. So thank you very much, Gauri and MassBio, for inviting us to present here today and to put antimicrobial resistance on the agenda. We think that's an unrecognized but extremely important topic. So my name is Milan Zdravkovic. I'm head of R&D and Chief Medical Officer here at Sniper Biome. And the title of my talk is going to be Reinventing Antimicrobial Agents Using CRISPR technology. So the lead program that I'll be discussing is supported by CARB-X. We're very grateful for the financial support that we have been getting, but also for the intellectual contributions. I think it's fair to say that our program is stronger, also with the input from CARB-X and their advisory boards. So a few words about who we are. We see ourselves as leaders in pioneering the use of CRISPR to target prokaryotic cells, and we have a very strong IP coverage. We're the first company to dose humans with a CRISPR therapeutic orally. We're led by a very experienced management team that has history in bringing drugs to the market, but also developing new companies. We have an R&D pipeline that is focused around addressing unmet medical needs, and I'll return a little bit more to that. We're backed by a very strong tier of venture capital, including the Lundbeck Foundation, Wellington Partners, Northeast Family Office, and what used to be LSP, and which is now EQT. So this slide gives an illustration of our pipeline. Fundamentally, we have two different modalities. We can either kill bacteria. Those are illustrated in the blue and the green, antimicrobial resistance and oncology. Or we can do what we call gut-directed gene therapy, whereby we can introduce synthetic biology into the existing gut microbiome and express therapeutic proteins, domain antibodies, or change the metabolic profile. The focus of my talk is going to be in the blue part around antimicrobial resistance and our programs targeting E. coli. We have some exploratory antimicrobial programs at the very bottom of the slide, looking at Pseudomonas klebsiella C. diff and also bacterial vaginosis. So I think it is probably not hidden to anyone that antimicrobial resistance is on the rise and is expected to be even more important in the future. This study is from the recent Lancet paper illustrating the significant morbidity and mortality associated with antimicrobial resistance. And as you can appreciate, E. coli is one of the main culprits and is also our initial, you can say, target focus. So this slide illustrates our technology. On the left-hand side, we fundamentally use CRISPR to selectively kill bacteria. But as in any other therapeutic modality, we need to have it delivered into the bacterial cell. We have two fundamental approaches we can use. We can either use a phage-based approach, so load our synthetic biology into a CRISPR or into a phage. We call it CRISPR-armed phage, and thereby elicit the killing we want to achieve. The other approach we can take is conjugation, whereby we can synthetically create a plasmid that can then be transferred into the bacterial cell and thereby kill the bacteria. The advantages of our approach is that it's highly selective and we can program it towards any species we want to. Since it's selective, it also spares the resident microbiota. We are using replicating phages, which means that they would amplify at the site of where the infection exists. But once the infection ceases to exist, the amplification would also stop. We have engineered our CRISPR systems so they may also be active on non-growing cells, such as biofilms. And we use Cas3 because we think that's a more effective way of killing the bacteria, unlike some of the other Cases. Our technology can be dosed both orally and intravenously. And to the right, you can see some of the applications that our technology may be used for, antimicrobial resistance, infectious disease, and of course, other therapeutic modalities. So this slide shows our lead program. We are targeting very vulnerable, high-risk hematological cancer patients that are at high risk of febrile neutropenia. They would traditionally, when their risk is highest, receive off-label fluoroquinolone to coach them through this period and prevent bloodstream infections from occurring. A fraction of these patients actually harbor fluoroquinolone-resistant E. coli, which fluoroquinolone does not protect them against. And that patient population has a very high risk of bloodstream infection, associated morbidity and mortality. So the way we are using our approach is that we see it as, you can say, combined first line, our CRISPR therapeutic plus fluoroquinolone to eradicate both the fluoroquinolone susceptible, but also the non-fluoroquinolone receptible E. coli. There's no approved therapy today for this patient population. We think there is an unmet need, and it's to a certain extent validated by the fast track designation or CARB-X support, and also the potential for a limited pathway within the FDA. The addressable market could be somewhere between $250 to $1 billion. Our first-in-man study is ongoing. We actually just completed enrollment, and phase 2 may start in 2023. So very briefly, our therapeutic modality for CRISPR phages to cover the totality of the E. coli biology. The idea is we want to kill the E. coli, in particular fluoroquinolone-resistant E. coli in the gut, and thereby hinder their passage into the bloodstream and thereby prevent these vulnerable patients from getting a bloodstream infection at the time where they're most vulnerable, typically associated with their hematopoietic stem cell transplantation. A few words about the technology and how we engineer. Typically, when we want to go for a target, and it's phage-based approach we take, we screen, you can say, phages in a number of different environmental sources. We try to screen them against a panel of bacteria. We have at least two orthogonal engineering approaches. We can either apply CRISPR, as well as, you can say, tail fiber engineering, the docking stations that the phages use to dock and carry the cargo into the bacteria. We load that into the wild-type phages. We do additional screenings, both for the manufacturing and efficacy and sustainability and ability to pass the gastrointestinal tract or IV. And then we come up with a final, you can say, cocktail of phages, and to cover E. coli, as I mentioned, we have 4 CRISPR-armed phages. A few words about some of the engineering we have done. One of our phages, alpha-15, mainly infects through LPS, and that tail fiber, there is a risk of resistance with LPS, so we have actually transferred another tail fiber onto our alpha-15 construct, TSX, and that means that there is a lower risk of resistance. I will share that on the next slide. As I mentioned, our CRISPR-Cas systems is also designed to target bacteria in biofilms. On the left-hand side here, you can see the in vitro activation of our CRISPR-Cas systems occurs within 15 to 30 minutes. On the right-hand side, you can see the wild-type, the construct alpha-15 versus our engineered construct alpha-15.2, and a much lower development of resistant strains, given, you can say, our combined engineering effort. As I mentioned, we can selectively target bacteria. This is illustrated here. On the right-hand side, you can see we're targeting E. coli. On the left-hand side of the slide, you can see that closely proximal neighbors of the E. coli is not susceptible to our constructs. As mentioned, we are going for hematological cancer patients at high risk of febrile neutropenia. Our initial 4-phase cocktail was screened against, you can say, a native panel of E. coli. And before we went into the clinical setting, we wanted to reassure ourselves that the host range that we saw, around 90%, would also be translatable into the patient population. And this is what I'm illustrating here. We sourced around 400 strains from patients that had been at a hematological cancer ward and had had an E. coli bloodstream infection. And we saw a coverage of our 4 CRISPR phages to the tune of around 90%. And these patients were from the past 3 years across Europe, US, Latin America, and Asia. What is maybe even more important than the broad coverage in general is also that when we look at fluoroquinolone multidrug resistant ESBL, but also carbapenem resistant strains, we also see a very high host range coverage to the tune of around 90%. We recognize, you know, that the numbers with carbapenem are low, but nevertheless, we are encouraged by, you can say, also the orthogonal data outside the broad E. coli biology. We have strong in vivo data. This is a preclinical study where we have dosed strep mice with a strain of E. coli, and we can achieve approximately a 4-log reduction, which also documents, you can say, the stability of our constructs to actually go through the gastrointestinal tract. We have a strategic collaboration with a manufacturing organization, one of the leading phage manufacturing organizations in Slovenia called Jafrel. We're very excited about that collaboration, and also I think the significant skill and capacity that Jafrel brings to our collaboration. Obviously, we have a master cell bank for production and also phage seeds. We're running it at a 30-liter full scale. We have good stability data, and we have actually not had a failed clinical batch. This slide shows the design of our clinical study. Before we went into a more vulnerable population, we wanted to reassure ourselves about foundational safety and also a pharmacodynamic effect in healthy volunteers, and I will return a little bit to that. So it's a classical dose escalation study. The healthy volunteers are dosed for 7 days, twice a day, escalated from 10 to the 8 to 10 to the 10 to 10 to the 12. Beyond safety and tolerability, we are looking at recovery of our phages in the gastrointestinal tract, and we are also looking at the ability of our CRISPR phages to actually kill E. coli in the gut of these patients, and we are doing that by enumerating E. coli, but also through metagenomic analysis. As I mentioned, we have completed dosing, and we are anxiously awaiting the results and expect to have unblinded day 35 data in mid-February. There's an additional 6 months follow-up that will be done subsequent to that, more to look at long-term safety. So I've talked about oral administration of 001 to prevent or to kill E. coli in the gut and thereby prevent its translocation into the bloodstream. We have a sister project where we also can or actually use 001 to treat difficult to treat E. coli infections. Here are some preclinical data that we can actually eradicate E. coli, both blood, kidney, spleen, and to a certain extent also in the bladder. This is with a antibiotic sensitive strain, but we are doing additional experiments also with non-sensitive E. coli strains to further augment our data package also in this space. So briefly, on the Series A, we think we have achieved significant milestones. We have a strong team, strong IP. We have validated our technology through collaborations with CARB-X, Novo Nordisk within gut-directed gene therapy, and MD Anderson Cancer Center within immune oncology. 001, our lead program, is progressing well in the clinical space. And while we have started within the E. coli biology, our technology essentially can be applied towards a number of other pathogens. Could be Pseudomonas, could be Klebsiella, could be bacterial vaginosis and maternal health. And our gut-directed gene therapy can actually also be used to express vaccines. I think with that, that was my last slide, and I'm happy to take any questions. Do you want me to stop presenting, or how does it work?
Yeah.
Yeah, okay, I'll do that.
Thank you, Milan. That was
Thank you.
fantastic. Great to hear about your technology. I actually have a question. So
Yes.
do you think it could be potentially applicable to biofilms where you have mixed colonies of organisms?
Yeah, so we have designed some of the synthetic biology, the way we have been working with it, to see if we can penetrate or actually also have it active in biofilms where the cells are more dormant. So we have tried to do that, and we have some in vitro data that supports it. I think at the end it's going to be, you know, what does the in vivo situation look like? But I think there are also situations actually in a gut environment that mimics a biofilm situation. So that was why we tried to engineer these aspects into it. But I mean, I think we're going to be much wiser also when we get the clinical data and see to which extent we actually lower E. coli. So I mean, we're a little bit anxious about that. Or yeah, excited, I would say. Yes.
Yeah, it's good anxiety, right?
Yes, exactly.
You have a great technology, great platform. Very exciting to see CRISPR being used to treat against bacteria and an AMR. So thank you for presenting this technology.
Thanks for inviting us.
It's wonderful, and thank you for calling in from Copenhagen. I know it's rather late for you.
No, welcome. So thank you very much. I'll put myself on mute, and then yeah, we'll take the next.
Thank you. And now it's my pleasure to introduce Mimi Yan from Phage Pro. Now this is a local Tufts spin-out that I'm proud to present here. Thank you, Mimi, for joining us, and glad to hear your presentation on yet another phage-based technology to treat AMR.
Great. Thank you so much for the intro, Gauri, and it's a pleasure to be here. Thank you for the opportunity to present our technology and our company to you all today. Like Gauri said, we are Boston-based. I'm actually in Chinatown right now in one of the phone booths at Tufts Launchpad. We are a spin-out out of Tufts University, and I'm excited to be able to tell you all our story and what our philosophy in phage is about. So let me just go ahead and start sharing my screen here. 1 second. All right. So I am CEO and co-founder of Phage Pro, where our mission is to develop innovative phage-based solutions to help the world's most vulnerable communities. So as many people have said today, phages are natural viral predators of bacteria. They've been used safely for the past 100 years to prevent and treat bacterial diseases. So they were discovered in the 1920s, used for multiple diseases, including cholera and other enteric diseases. And ultimately, phages were abandoned when, as Martin mentioned earlier today, antibiotics came on the scene in the 1940s. And as you see here today, there's been this recent renaissance in phage therapy here as one of the viable solutions to antibiotic resistance. And we in the US and Europe are starting to build out some infrastructure to support the commercialization of this renewed modality. At Phage Pro, we believe that we need to think even more globally, more interconnectedly, and holistically to truly address the problems that we have with AMR and try not to repeat some of those mistakes of the past. So we are passionate about a phages for all ecosystem to solve the antibiotic resistance crisis in not only in a therapeutic way, but in a preventative way as well. So why do we adopt this philosophy? And as it has been reiterated before today, It's because AMR is going to affect our counterparts in Africa and Asia the most, especially in resource-limited settings where communities are more vulnerable to infections. So this map is showing the proportional number of deaths from antibiotic resistance that will happen in 2050, where light blue is the lowest mortality rate and purple is the highest mortality rate. And as we know from the ongoing COVID pandemic, infectious disease really reveals the cracks in our global healthcare infrastructure. And it also illuminates how critical it is we act as a global community to address these issues. So what happens in Africa and Asia undoubtedly affects us here in North America and in Europe. And we need to start thinking about how to build up a flexible and adaptable infrastructure and pipeline of alternative antimicrobials that work as prophylaxis and as treatment. So when it comes to antibiotic resistance product development for resource-limited settings, particularly in low and middle income countries, PhagePro thinks of 3 critical factors. So it has to be specific to not add additional evolutionary pressure to current antibiotic resistance mechanisms. It has to be easy to distribute. So anything that would make it more convenient for either the patient, the healthcare worker, or the government organization responsible for healthcare delivery. And examples of this would include independence from the cold chain, or exploring different routes of administration to reduce the burden on healthcare workers, as well as increase patient compliance and buy-in, especially with a newer modality or a renewed modality like phages. And last but not least, it must be affordable. So this describes the individual doses themselves in terms of their pricing, but from a public health and a prophylactic standpoint, as well as the cost of the campaigns. So at PhagePro, we think a lot about the economics of prevention versus treatment, since investing in prevention will be ultimately more cost-effective, both in terms of finances and human lives for that particular community or government. So phages hit all 3 of these criteria, and as a team, we evaluated our strengths not only in the scientific knowledge, but in country partnerships as well. And that's something I'm going to reiterate throughout this whole talk, that without partnerships, a renewed modality like phage would not be able to be sustained in the future, but also working in emerging markets, we need in-country partnerships for that to actually be viable. So we are a spin out from the Camilli Lab at Tufts University, like I mentioned before, and the Camilli Labs works on cholera and pneumonia. And given the historic beginning of phage therapy in enteric diseases with demonstrated success for that time, we're currently focused on using phages as a prophylaxis for cholera. So cholera, for just a gentle reminder, is an acute, severely dehydrating diarrheal disease, and it can kill within 12 hours of symptoms appearing. The WHO estimates that there are 1.3 billion people in the world at risk for cholera, and without rehydration therapy, the fatality rate is approximately 40%. Approximately half of those deaths are in children under 5. And this is largely due to infrastructure issues in hygiene and sanitation, because the primary mode of transmission for cholera is water contamination. So for example, this is a picture of a toilet that I took in a cholera endemic area where the waste flows into a nearby stream where the community gathers its water. What has been increasingly recognized, though, however, in enteric diseases, in AMR, but particularly in cholera, is that household transmission plays a huge role. So for cholera, 80% of secondary cases during an outbreak are actually due to person-to-person transmission, and it's quite rapid. Household contacts will come down with cholera symptoms within 2 to 3 days of the index case, and that just spreads through the community like wildfire. So when Haiti was first hit with cholera in 2010 after the earthquake, cholera spread throughout the whole country in 1 week. People know how to protect themselves, they know about the disease, but at the time, they're not given a lot of means to do so. And this is where we think phages can really help. So our phage cocktail, which we call prophylactic VC or PVC for short, includes 3 distinct vibriophages, ICP1, ICP2, and ICP3. And there's distinct receptors for the virulence factors of cholera. ICP1 even has its own CRISPR-Cas system. So it uses a classic mechanism that bacteria use to protect themselves against phage, against the bacteria itself. So extensive characterization has been done genomically and microbiologically to understand these 3 phages and thoughtfully utilize them as tools. So because of that, we can confidently say that PVC can provide immediate protection, which is greatly needed for rapid intervention measures to stop household spread, and it's specific for cholera bacteria, so it doesn't contribute to even existing mechanisms of antibiotic resistance, which is an increasingly pressing issue. I don't know if you all have heard in the news, we have now world-breaking cases of cholera all around the world in places where cholera was thought to be eliminated. And what we're hearing from our clinicians on the ground as well is that extremely drug-resistant or XDR strains of cholera are being identified. And what's scary about this is that there is a clonal nature to this disease. So what spreads in one area undoubtedly spreads all across the world. From a phage perspective, that actually makes it a little bit easier to get coverage, but certainly from a disease perspective, that just brings more urgency to this epidemic. So the last thing I'll talk about here is that the ideal formulation that PhagePro is working towards is an oral tablet that can be self-administered, and it does not need to be kept in the cold chain, so that storage and distribution are simple. And again, this is incredibly important to be stable in hot and humid temperatures for global health product development, but in general as well, we've seen it play out for current storage requirements for COVID vaccines. So we are thinking a lot about the integration of the context for the distribution of that product. And we really do think it's of equal importance sometimes to the science behind the product itself. So cold chain infrastructure is simply not reliable in places where people are dealing with cholera epidemics. And often the burden for that cold chain storage is shifted to community health workers who are on the front lines. So knowing all of this, talking to our stakeholders, talking to our partners, this has pushed us to innovate earlier. So PVC is currently already being developed into a stable oral solid dose formulation so that it can actually be of use in preventing cholera outbreaks once it is available to our stakeholders and communities. So to show a little bit about what we think the power of PVC could be, we tested it in proof of concept studies in animal models. This is using a liquid formulation just to show proof of concept in vivo. We showed it in 2 animal models of cholera, the infant mouse model and the infant rabbit model, but here you're seeing the infant rabbit model data as it's the gold standard in the field. These animals also experience the same symptoms of cholera as humans do. So here in this rabbit experiment, we orally administered a liquid formulation of PVC, waited 3 or 24 hours, then challenged the animals with Vibrio cholera orally, and monitor the animal body weights and clinical symptoms until the endpoint of that experiment. So in black, we can see the group that did not get PVC. Within 10 to 12 hours, the animals lost between 10 to 20% of their body weight due to diarrheal symptoms. For the PVC treated groups, both 3 and 24 hours, they were much healthier and protected. So this gave us the confidence that we have something that could be compelling, could be potentially transformative for the cholera prophylaxis toolkit, and add to current methods of prevention and outbreak control. So if we do think about what is going on in the preventative market for cholera, we're looking at a large market, but ultimately thinking about who is actually accessible to it. So what we've done here, because cholera is a little bit different in terms of surveillance, it's a little bit more chaotic, I would say, than other disease indications. We decided to use the oral cholera vaccine as a corollary for our market analysis. So thinking about that, there's the endemic cholera market as well as the travel medicine market. So we have these 2 shown here. Ultimately, we think that the combined market for both of those is about 217 million. Additionally, cholera is a part of the Tropical Disease Priority Review Voucher Program from the US FDA, and recent sales of that voucher has been approximately $100 million. So creating more incentive for us to work on these diseases that are really diseases of poverty, but also there is some financial gains as well as a lot of social gains to be had when we work on indications such as this. So if we think about competition, I think for us, antibiotics remain the main competition for cholera work, for enteric disease work. it's something that the communities are very comfortable with and have a lot of access to as well. Prescriptions are usually not required for access to antibiotics, so that overuse has contributed to a lot of widespread resistance. And it's not only jeopardizing the sustainability of antibiotics for cholera, but for enteric diseases in general in that area, since cholera doesn't exist solely in the ecosystem by itself. It exists with a lot of other bacteria and allows for transmission of AMR genes to other bacteria as well. So antibiotics, patient buy-in, all of that could be barriers to having our phages be effective in the communities and the marketplace. In terms of other phage competitors, so far we are the only ones who are commercializing cholera, at least, a cocktail for cholera, but there are certainly other phage approaches in the pipeline targeting a lot of other indications, as you've heard today. Most of approximately half of them have natural phage cocktails. Some of them have engineered phage cocktails, and also others are approaching it from engineered endolysin approaches as well. And they're targeting mostly Pseudomonas, E. coli, Klebsiella, Staph aureus, C. diff. So there's a lot of people in the ecosystem to learn from. But currently, right now, we are the only ones really focused on using phages against cholera. So the reason I mentioned that is because I want to talk about what PhagePro can do in the future using a lot of this technology that and a lot of the relationships that we have gained in using phages for enteric diseases, because on the WHO and the CDC priority lists, the ones that are highlighted in yellow are the ones that phage companies have been working on so far, but there certainly leaves a lot of other opportunities for PhagePro to go after using our expertise and also our relationships with in-country partners and collaborators to collectively take what we've learned from cholera so far and accelerate that product development for other diseases that are high burden, both in LMICs and emerging markets, but for high-income countries as well. And one of those opportunities could be for Shigella. There's 80 to 165 million cases worldwide. It's the second leading cause of diarrheal mortality among all ages, and there's no vaccine available. And currently there is a phage trial being done on Shigella, but we're thinking about it again from a prophylaxis perspective as well. How do we prevent shigellosis from occurring? The other indication that would be amenable as well is Helicobacter pylori. There are 3 billion, and I believe this is an underestimate, 3 billion people in the world infected with H. pylori. It causes gastric cancer, which is globally the third leading cause of cancer-related death. Again, no vaccine is available, and we do think that a lot of what we've learned for cholera and for access in LMICs in particular can contribute to this PhagePro blueprint to go after these indications. Because ultimately the way that we think about it is an integrated and iterative product development. So taking a lot of the classic pieces of biotech product development and thinking about it not only in a nuanced way for phage to address ultimately phage resistance that will pop up, but also thinking about it for global health. So we are thoughtfully engaging with organizations, regional and global, to build out the foundation to support phage therapy in this manner. So it's not only the science, but also the partnerships, the relationships to understand context and integrate that early into product development to ensure that not only will it work medically, but will also be used. And the other aspect of this that I talked about with resistance is building out that phage therapy ecosystem that allows us to invest in communities that have been harmed by lack of equity and access and build capacity in emerging markets in a way that synergizes with traditional product development in biotech so that we can allow for quick iteration in those regions when phages inevitably will need to be switched out. Because ultimately all of this needs to be sustainable on a global scale. And PhagePro has been doing the work in these complex and rich environments with very passionate collaborators to make sure we, and more importantly, they are not being missed out on this opportunity. And what that means is investing in horizontal approaches to health. So a little bit different from what we talk about in biotech, but this is language borrowed from global health. So instead of being super disease focused, horizontal approach is thinking about how it could think about infrastructure and capacity building, and we really think that could synergize very well with product development. So that's the way that we're thinking about growing our blueprint and our company in the future. And ultimately, why the urgency? All of this, I presented that figure at the very beginning, but I wanted to reiterate again that I think we in infectious diseases well know about the burden of AMR, but the Wellcome Trust had called it a silent epidemic before. But it really isn't silent anymore. It's been declared by the WHO that it's a top 10 global public health threat. It caused more deaths than HIV or AIDS or malaria in 2019. Again, that highest burden is in Africa and South Asia, and phages really are a viable alternative. So we need to start investing in this infrastructure now so that all of these new solutions, new modalities that we're talking about today can remain sustainable. And this is something that we've been working on since 2018 when we were only 2 co-founders. Now we have 2 NIH grants with 5 full-time employees and established partnerships in the US and abroad, and we're excited to continue growing. It was just me and Professor Andrew Camilli at the beginning building this out, but out of our 5 employees now, I'm very proud to say that not only are we working on having equitable partnerships externally, keeping in mind global health power dynamics, but also internally as well. So across all levels, from research associate to our strategic advisory board, we're thinking about DEI and thinking about incorporating that as a philosophy for our company and for our culture. So I can proudly say that out of our 5 full-time employees, our team is 80% women and 80% BIPOC. And we're guided by an incredible strategic advisory board comprised of experts in biotech, phage product development, and we're currently recruiting for more clinical, regulatory, and global health experts as well. So thank you so much for listening to me today. I hope you join us in our fight. I'm happy to take questions, or if we don't get a chance to chat today, please feel free to ping me on LinkedIn, Twitter, or send an email. I'm always happy to chat. So thank you so much.
Thank you, Min. That was fantastic. Great to see a young founder like you, launching this company. What advice do you have for young startup founders in this space?
Oh gosh,
Your own experiences.
I think asking for help as early as possible. I think a lot of what gets in the way of young startup founders is feeling that imposter syndrome, feeling like we're a little bit out of place among all of these experienced biotech experts and startup founders here in this ecosystem. And sometimes that can be a little bit intimidating. And for someone like me, where it's not only youth, but also I'm a woman, I'm Asian American, sometimes there's a lot of times where you can feel out of place. And I think there are people there for you. There are people rooting for you. You just have to ask for help. And once you find your community, you'll be able to push your passion project forward. Yeah.
I see that you went through Mass Connect. So do you think you're, going through the program, being part of the MassBio community is up?
Absolutely. So that's funny. 3 of my advisors are from MassBio. So 2 of them are from my Mass Connect program, and Alice is on the board of MassBio. So the program launched us in 2018. It was an incredible opportunity for me. So thank you to MassBio. Thank you to all of you coordinating this day, as well as for all the people who have been working on this for the past few years.
Yeah. So I do have a question coming in from the audience.
Sure.
Can you speak to how you built your in-country stakeholder networks?
How do I speak to them?
Can you speak to how you built your in-country stakeholder network?
Yes, absolutely. So one of our most important partner is the International Center for Diarrheal Disease Research in Dhaka, Bangladesh. They are world renowned for doing leading work in cholera and other diarrheal diseases as well. The way I got to know them is through Professor Andrew Camilli, who had been working with them for the past 20 years. And I've established my own relationship with them through conferences, through visiting, through really getting to know them and their strengths. And for the other in-country partnerships that we have established, Again, it's going to conferences and networking. So a lot of those people are normally not invited to conferences. They're normally not present as speakers. They're either not invited or not included. So sometimes it does involve going to the country itself, finding just one point person and just having a bunch of coffee chats or Zoom chats or whatever it is to start to establish a foundation on the ground and some footing on the ground. But I think ultimately it came down to a lot of people connections because conference connections are a little skewed also in distribution.
Yeah, fun fact, my postdoc lab was right next to Rita Colwell's lab, and she's a pioneer in Vibrio,
Yeah.
and she was on the ground in Haiti collecting samples during the cholera outbreak there. And I think her lab was very active in Bangladesh and other places where there have been outbreaks. So, and every weekend they would autoclave oysters, and the lab floor didn't smell that great because of isolating Vibrio.
Yeah.
So that's about it from us, and I will be sure to connect you with others that have questions. And I'll be introducing the next presenter now. But if any questions come in through the chat, I'll definitely connect you to those folks, and you can directly interact with them. But please welcome to this Innovation Week Infectious Disease Day. Great to hear your presentation, and let's continue to stay in touch and see how we can help you.
Absolutely. Thank you again for the opportunity to speak. Have a good day.
You too. So now I'm going to introduce the next presentation. This is pre-recorded because this is a company that's based in India, and really this wasn't, the time difference was a little much. I'm going to introduce Bugworks Inc. It's based in India. They have a novel platform for developing anti-infectives. And the speaker is Bala Subramanian, who's the COO and the co-founder of this company.
Hi, my name is Bala Subramanian. I'm the co-founder and COO of Bugworks. I thank MassBio for inviting me to present on behalf of Bugworks at the Innovation Week in December. And what I'm going to share with you is a journey of a discovery of a novel broad-spectrum antibacterial agent. And before I begin with the science presentation, let me thank the funders profusely, the public funders, Carbex, who's been probably the most generous for us in terms of supporting this program, BIRAC from the Government of India, in whose facilities we are housed, and GARDP, who's supporting our clinical program at this stage. And definitely not to forget our private investors who have supported right through our journey over the last decade. With that, let me introduce our clinical candidate, BWC0977. And that's the compound which I'm going to focus in today's talk. This belongs to a class that we've named Gyrox because they are DNA gyrase inhibitors. It's a novel broad-spectrum antibiotic. We believe the first of its kind in terms of the spectrum, probably in over the last 5 or 6 decades. It has the potential to cover a variety of indications covered by a whole spectrum of pathogens, such as complicated UTI, complicated intra-abdominal, hospital-acquired bacterial pneumonia, ventilator-associated bacterial pneumonia, cystic fibrosis, biothreat, warfighter infections, and a whole slew of indications. Basically, it addresses a lot of unmet needs and a lot of gaps in these indications, because even though there have been a flurry of recent approvals, they don't cover all of the gram-negatives and certainly not the gram-positives in the recent approvals. Currently, 977 is progressing through a phase 1 study, which will enable us to assess its therapeutic index because we will get the tolerability and PK data from the phase 1 study. We believe largely it can address 3 different TPPs. The holy grail for us is an intravenous plus oral step down, which covers both critical care and community and biothreat infections, because it has a tremendous competitive advantage because it covers the entire gamut of enterobacterials, the non-fermenters, the biothreat pathogens, etc. And as I said, there has been no recent approvals in terms of having both an IV and an oral step down. The second TPP is if we have challenges in the IV, then we're looking at an oral, clearly looking at all non-ICU based indications. And in the last decade, there is no oral new drug in the market. And the last is an intravenous only, where clearly focus would be the ICU based indications because there are gaps in the recent approvals in terms of non-fermenters and the key pathogens implicated in pneumonia. Our clear focus at this point is TPP1, IV plus oral step down, and that's what we are progressing at this point. So I'm going to spend the next few minutes, the remainder of the talk, perhaps in terms of the actual data on this compound, the exciting data as to why this is a novel and an exciting new opportunity. This is a dual target inhibitor, which inhibits both DNA gyrase and topoisomerase 4. If you look at the top left in terms of the gel, where you're looking at a DNA supercoiling activity with the E. coli enzyme, it's an incredibly potent at about 5 nanomolar inhibition. Comparison, ciprofloxacin, which is probably one of the most potent fluoroquinolones, is about 200 nanomolar. And if you look at the left bottom, which is looking at the topo4 inhibition in terms of a decatenation activity, again, it's about 10 nanomolar potency, whereas the ciprofloxacin, the fluoroquinolones, actually have an extremely weak decatenation activity. So primarily, these are dual target versus the fluoroquinolones, largely a biased DNA gyrase inhibitor class. The difference doesn't stop there. If you look at the modeling information in terms of the binding sites for the compound vis-a-vis ciprofloxacin in this case, as an archetypal of the fluoroquinolone class, the binding residues are actually very different, both in the gyrase and the topo4. And this then, we've used this information to confirm that they're actually non-overlapping in terms of resistance. Before I go to that, let me also explain another key difference with the fluoroquinolones. So if you look at the top left, what you can see is in this, as you increase the concentration of the inhibitor, there is actually an increased amount of nicked DNA species, indicating there's a single-stranded break which is stabilized. As opposed to the case of the ciprofloxacin, it's the linear DNA which keeps increasing as you increase the drug concentration, moving from left to right, or rather right to left. And what you see is that is because it stabilizes a double-stranded break. This has implications in terms of the SOS repair systems, etc., but I won't go there. What we're showing in the bottom is engineered mutations based on the modeling information. So if you look at the bottom left, the enzyme which is created with the mutations conferring resistance for ciprofloxacin does not confer resistance to 977 and vice versa. And the bottom right is if you take these engineered enzymes in a bacterial background, actually mutant strains, you require the double mutation in the gyrase and the PAR-C to confer resistance for 977, again indicating that these are dual target and you actually need to inhibit both. Perhaps the significant aspect of that is that all our attempts to generate resistant mutants in the lab, whether it's by standard one-step antibiotic exposures or serial passages, or even using hypermutatable strains, what you see is that we have been unable to raise mutants in the gram-negative agents. And we infer the resistance frequency to be less than 10 power minus 9, thereby indicating that this has a tremendous potential in terms of a very long life in the clinic before it actually generates clinical resistance. And because of the dual target, very highly potent on both the targets and across the entire spectrum of the gram-negative bacteria. So that brings me to the MIC distribution. Here I'm showing some key pathogens. If you look at the left-hand panel, basically the distribution across the key pathogens of the non-fermenters, Pseudomonas acinetobacter, E. coli, and Klebsiella. The MIC90 distribution is beautiful, anywhere between 0.5 to 2 microgram per ml. And this MIC90 holds good if you look at the MDR subsets on the right side, pretty much better than any of the comparators in most cases. But more importantly, the MIC90 is retained whether these are susceptible isolates or drug-resistant isolates. We didn't just stop with these 4. We've actually gone to town, tested probably about 10,000 isolates now across a whole host of gram-negative and gram-positive pathogens. Beautiful MIC90s, which we believe we can actually achieve in terms of therapeutic exposures. This is again a reiteration in terms of the retention of the MIC90 against the MDR subset. This is work specifically done through Carbex, and it was generated at IHMA, where you can see the top row where the 977 MIC90s are retained, irrespective of the multi-drug resistance phenotype that you see against all of these about 2,000 isolates across these gram-negative pathogens. This is why this is an exciting broad spectrum opportunity. And if you look at, there are 2 other NBTIs which are ahead in clinic in the development, Gepotidacin, which is probably going to go for approval very recently in the near future, that you see that some of the key pathogens in the top in terms of the Enterobacteriaceae, the non-fermenters, and as well as some of the key gram-positives, Gepo and Zoliflo do not cover. 977 touches all of them. This is why this is such an exciting opportunity. This is also an exciting opportunity because it actually provides a tremendous stockpiling opportunity because it works beautifully against all of the bioterrorism agents such that it potentially becomes a one-stop solution. Not just MIC, we've actually confirmed them in a series of in vivo experiments looking at oral efficacy against these pathogens. These are very early studies against these biothreat pathogens. All it's telling you is that this has tremendous potential as an oral option to actually use as stockpiling. Of course, these are very long-term studies. If you look at it, these are 33 days, 45-day studies. So these studies are actually just progressing in terms of walking through what the optimal dose that would be in terms of actually getting to address the biothreat stockpiling. I'll come back to the efficacy in a minute, but before that, let me walk you through in terms of its ADMET profile. It's got about 6 to 10% protein, free in terms of the protein binding, anywhere between 85 to 95% bound. It's based on its, if you look at the hepatocyte clearance, it's considered to be moderately cleared compound, about 28 liters per half per kg in terms of the liver blood flow, and clearly doesn't touch any of the CYP enzymes in terms of inhibition or the induction. And if you look at multi-species in vivo PK, it beautifully has linear pharmacokinetics across species and dose. There is no evidence of partitioning into either RBCs or the central nervous system. It doesn't cross the blood-brain barrier. There is no accumulation seen on multiple dosing in preclinical species. No major metabolites formed in the dosing period. It is a substrate for 2 renal transporters, and this may be the reason why we are actually seeing quite a bit of unchanged compound in the urine over 24 hours in both the preclinical species. It is not a substrate for any of the efflux pumps like PGP, BCRP, etc. It's also got great oral legs. This is an dose escalation pharmacokinetic profile from a dog study, where what we can see is that the compound behaves nicely, very nice linear range, all the way up to 300 milligram per kilogram, and about 50% bioavailability based on the dog IV data. Of course, there are antibiotics which are probably at 90% bioavailability, but given its spectrum, 50% bioavailability is actually still a good place to be, which we will exploit as an IV plus oral step down. The other interesting aspect of this compound is that it beautifully partitions in the epithelial lining fluid in the lungs. This is data coming from an infected rat from a neutropenic lung infection model. And what you can see is that the 977 has significantly higher levels in the epithelial lining fluid as opposed to the free concentrations seen in the plasma. Why is this important? This would be a tremendous opportunity in terms of looking at pneumonia indications because of its preferential partitioning in the epithelial lining fluid. So here I'm going to quickly in one slide sum up all of the efficacy data that is available. We've actually studied this extensively in a variety of models. What I'm showing you is a non-typical model, which is a rat infection model. And the reason for that was that it took us a long time to actually optimize the oral. And so by the time these studies were done, it was much easier to do the IV route. And the bolus injection is not suitable because we could not get a dose escalation. And the only way to tackle this was 1 hour to 2 hour infusions, and the rat became a very suitable model. The rat is also very advantageous because it's the same tox species and the protein binding values are identical between rat and humans. So there's a lot of translation that is easily possible from this rat model. The infection kinetics in the neutropenic rat model is very similar to what you see in the mouse model. So there's actually quite a bit of extrapolation of what can make. Long story short, if you look at the dose fractionation studies basically told us that the AUC over MIC is the key PK/PD driver. And basically summation of a variety of strains across 4 different pathogens. What you see is that target AUC over MIC of about approximately 70 drives that 1 log kill efficacy, which will be the guide for us as we move into clinical development. This is what we will look at in terms of what we need to achieve in our SAD and MAD study to actually look at how we can position it subsequently in a phase 2 study. We've also completed an extensive GLP tox profiling, and we looked at both rats and dogs. We basically carried out 14-day repeat dose studies. But before I go to the 14-day studies, what I want to point out is that as an NBTI class, one of the first things that is always a red flag is the cardiotox risk. And what we did was, based on all the preclinical evidence, moved on to the conscious dog telemetry. While we did see some small variable changes at approximately 16 to 15 times above the therapeutic Cmax, this was still considered good enough in terms of a novel window to move forward. The gyrase and topo4, because of the very nature of the enzyme inhibition that actually touches the DNA, the genotox risk is always there. And we did study both in terms of in vitro and in vivo. The in vitro is all clean. The in vivo micronucleus test was negative. The in vivo liver comet assay, we did see some slight increases, but these were variable and not considered positive. And the headroom was anywhere 10 to 20x in terms of the AUC and 25 to 50x in terms of the Cmax. From the GLP tox studies, the 14-day repeat dose tox studies, interestingly enough, across the low, medium, and high dose groups, we didn't see any major changes in the rats. It was actually quite a clean study. In the dogs, only in the liver and only in the high dose groups, we actually saw general tox evidence. And this was basically reversible elevation of liver enzymes. And based on the fact that based on the liver enzyme data, etc., the novel was determined and the more sensitive species was taken. And based on that, the margins were calculated about 1 to 3x in terms of AUC and 5 to 12x in terms of Cmax. Agreeably, these are tight margins, but most antibiotics actually live with such tight margins. These are short-term treatments, and we would always monitor in terms of reversible elevation of liver enzymes. The last of it is the secondary farm data. We've known from the secondary farm data that acetylcholinesterase inhibition could cause an issue. We did see minor reversible changes in terms of respiration, etc., in the preclinical species, and we clearly would be monitoring that very carefully as we move into the clinical program. So based on this, we actually progressed this compound in volunteers, and this was a study in Australia where the study is in progress. This is following a 2-hour IV intravenous administration, both in terms of a single ascending dose package followed by a multiple ascending dose package. I'm afraid I don't have any data to share here simply because the study is midway. We walked through successfully 5 dose cohorts. So far, so good. We basically have not seen major tox findings. We've not seen major adverse events, and the PK is behaving as we had predicted from the preclinical models. We are yet to commence in terms of the subsequent phases, and we're likely to finish all of this over the next 6 to 9 months. And so this is where we are. We do not have at this point data to share from the human study, but clearly these are exciting times, and based on what the blinded data that we have, we believe it has the legs to cover those key pathogens that we talked about in the earlier part of the presentation. That brings me to acknowledging all of the players. This is not possible just as a Bugworks team of barely 20 people inside the company. This is a very large family. We call it the global village. A variety of players, both all the way from stretching from San Francisco to the UK cluster, to Japan, to Australia, where the clinical trial is happening. Many of the folks that we work with are actually former colleagues from the same pharmaceutical company that we all came from, which is AstraZeneca. And we also work with a variety of universities, key opinion leaders, professors, such as Dr. William Hope Shampadas at University of Liverpool, Dr. George Rusano at University of Florida, etc., who actually helped build this entire package. And that is what we are now moving forward with. That brings me to the conclusion of my presentation. Thank you all for listening.
Hi, everyone. My name is Gauri Nair, and we are now on to our last presentation of the day. I will be introducing you to the J&J Spotlight presentation, which is Felix Biotechnology, coming in all the way from Toronto. They are a JLab resident, and proud to have you here as the J&J's Spotlight presentation today. I introduce you to Robert McBride.
Thank you so much, Gauri. And I'd just like to kick it off by saying thanks to the Blue Knight program and to J&J for allowing us to be here, and to MassBio for organizing this. My name is Robert McBride. I am one of the co-founders and the CEO of Felix Biotechnology. We are a company. We're actually based in South San Francisco, not Toronto, but we're in the JLabs ecosystem. I haven't had a chance to visit the Toronto site yet, but I've heard it's really great. What we do is we design viruses specifically to target and kill bacteria that are resistant to small molecules. And I know that the audience is very aware of what this challenge is, but the reason our reason for existence is specifically, let me figure out how to use this. So specifically to address this challenge. So it's a huge challenge. It's looming, it's getting worse. And at Felix, our focus is on making sure that we're doing everything we can to address this ahead of time, and to develop tools to add to our toolbox to make sure that the future without antibiotics is not a future that we have to face. And the tool that we're using to address that is the bacteriophage. This is something some of you may know something about. It's a virus that kills specifically bacteria. We've known about it for a long time, and it has a number of advantages over small molecules. I want to make sure that I'm very clear. This isn't a panacea, right? This is a tool, and we're trying to develop it so that we can add it to our toolbox, of which small molecules obviously is the prevailing tool. It has a number of advantages over small molecules, right? It is able to kill bacteria that are resistant to all drugs. It does have a very high specificity, which is increasingly appreciated. One of the things that I think is underappreciated is it has a very low toxicity when comparing it to other small molecules. It has a novel mode of action. You can use a small dose because it does self-amplify, and it can target biofilms. And one of the most exciting things from our perspective is that we have the ability, using this approach, if you design it well, to turn back the clock on the evolution of resistance to small molecules. A question that often comes up, and I think that is worth addressing upfront in this presentation, is if phage has been around for so long, why do we not yet have a phage product on the market? And that is something that Felix, when we were getting started, we thought long and hard about. And as far as we're concerned, there are three key issues that any company in this space need to be able to address in order to usefully deploy this as a tool to help people. And we're focused on trying to develop as generalizable and as durable a tool as possible. And the three key challenges that we're focused on is, most of the viruses that we are interested in and that we think have the best potential for patients are wild type. They're novel viruses from the environment. And as such, we don't have the traditional tools in place to modify them. And so we need to have a facile ability to manipulate and engineer the tools that we can, the phage that we are interested in from the environment in order to improve their phenotype and improve their patentability. We have a very exciting digital phage platform that allows us to do that with almost any phage. A second big issue is that phages have a very narrow host range, and quite often this narrowness of a host range is smaller than the phylogenetic diversity of the bacteria within an indication. And so you have to ensure that your phage therapy can kill the majority of the bacteria within a specific indication to benefit the patient. And that's really challenging to do. We have taken a machine learning approach to expanding host range. We're very excited about the progress that we've made in this area. And lastly, it's the facility with which bacteria become resistant to phage. So this is one of the known challenges. Bacteria can become resistant to phage fairly quickly. And at Felix, our approach is to design our viruses to target virulence factors on the bacteria. And these are essential aspects of the bacterial behavior so that if they become resistant to the phage, they then lose that virulence. And the sort of example we give the most is, bacteria is resistant to antibiotics, but if it becomes resistant to phage, it becomes resensitized to antibiotics. And so that's an example of that type of an approach. Our platform is really exciting. It feels kind of science fiction-y. So we get large data sets mapping a phenotype of interest to genetic variability within the virus. And we use a couple of different machine learning algorithms to identify what the genetic underpinnings of the behavior are that we're interested in. We then print those genomes out, assemble them, and boot them up into living viruses. And then we've got a very high throughput strategy for evaluating whether or not those printed viruses do what we think they do. And this is kind of our flywheel, right? So the more times we turn, the better we get, and the more we learn, and the better positioned we are to deliver phages that benefit patients moving forward. One of the unexpected benefits of this approach is, with regards to discovering new viruses, and I said most of the viruses we look into, and I think quite a few companies look into, are viruses that we've isolated from the environment that are novel. With this synthetic approach, we can now, in addition to mining sewage and environmental samples from around the world, we can mine digital information from around the world to identify viruses that have interesting variation related to the viruses that we have. And we can actually print those out, right? And some of those viruses may never have actually been isolated or examined in the lab. So this is an extremely powerful side effect of the platform that we have. We also have a very robust engineering tool in viruses that are not amenable to printing, or if we want to sort of very quickly make minor adjustments to one of the viruses that we have, and this is the CRISPR-Cas13a approach with counter selection that was developed in conjunction with Joe Bondi-Dedemy, has a number of advantages over the traditional sort of Cas9 approaches. And with the counter selection, we're actually able to modify lytic viruses fairly quickly. And we have very few limitations on the application of this technology in most of the wild-type viruses that we are working on. So moving on to our business model. So we right now have a hybrid business model. We are very focused on using our platform to make novel viruses in the antibiotic space. But we can also use that platform to make novel viruses for customers who are interested in targeted chemical-free solutions to bacterial challenges. And we have, partnerships right now, revenue-generated partnerships in the personal care, the animal health, and the skincare market. This generates revenue, but it also generates data. And the data is really critical for our ability to enhance our machine learning algorithms. And then our second sort of business model is to advance clinical assets to the stage where they de-risk sufficiently to allow us to out-license or co-develop with partners who have more experience in the particular space. And so our goal is to, as quickly as possible, get solutions into the hands of patients who can't wait. Clinically, we have a number of assets, and we work very closely and have in-licensed a number of these assets from the Yale team of Paul Turner at Yale. So the lead asset that we have in-licensed is in targeting Pseudomonas aeruginosa infections and cystic fibrosis, and I'll give you a little bit more information on that in a second. The second asset that we've been focused on and working on is targeting MRSA infections and prosthetic joint infections, and in a few other indications. And then we've got very early work in Mycobacterium, Acinetobacter, and Burkholderia, and those are primarily focused on CF applications. With regards to our first asset, though, one of the things that got us really excited about this, and this publication will be coming out shortly, and if anybody is interested in learning more, please do drop me an email after this, and I can share the preprint with you. But the lead asset was deployed targeting cystic fibrosis patients who had Pseudomonas infections under a compassionate use IND from the FDA. So the goal here is not to gather data, but data was gathered along the way. And it definitely suggested that this approach was working, not just in terms of its ability to improve lung function in these patients, but also to reduce microbial load. And to further examine this, the team at Yale got an investigator-initiated IND up and running. The idea was to treat 36 patients. We've treated 8 patients so far, and on the basis of the results that have been generated, this trial is now being closed and moving to open label, and we should have data out before the end of the year, hopefully, showing that in a controlled placebo-blinded environment, there is a significant phenotype demonstrating the efficacy of this approach, right, and substantiating the previous compassionate use data that was collected. So very excited. And again, for us, this is data that reduces the risk that this approach can work in a real world environment. We've got a number of different, strategies in the IP space. Our platform IP, we've got, IP around the ability to characterize and understand receptors, which is critical to our trade-off approach. We are keeping our machine learning approach in sort of the trade secret space. We've got the Cas13 IP from UCSF, and then we've got internally filed IP around building the synthetic phages. And then as far as our assets go, we have in-licensed assets from Yale, and then each of the phages that we modify are IPable. So this is kind of our IP approach. We are differentiated from others in the space in that we are really, really focused on durability, right? So our approach, we think, is optimized for chronic infections. All the companies on this slide are doing amazing work in the space. They all have slightly different approaches, and I think those approaches will work in different areas. And again, we're focused on the chronic indications, and we think we are significantly differentiated and will have a significant benefit, again, when we're treating repeatedly because of our ability to deal with the evolutionary outcomes of bacterial resistance. We are a small team. We're based in South San Francisco, as I said in the beginning. I'm the CEO. I have a technical background in PhD and postdoc in evolutionary microbiology, and have been in startups now for the last 12 years. We've got a number of scientists on the ground, and we've got a number of data scientists who are helping us with our ML and our bioinformatics pipelines. We wouldn't be able to do what we do were it not for the help of a number of really amazing, dedicated, and talented advisors. They're on the slide. I won't go into all of them. But obviously, we're super excited to have Jennifer on board, specifically when we're thinking about engineering viruses and the ability to target payloads to specific virus types. Paul's one of the co-founders, along with Vivek and Adam, and a few others. So really, really grateful and excited by the quality of the advisory board that we have. We are currently in the midst of a seed raise. We're raising 3.5. That'll give us 16 months of runway. Our pre-seed was 2.4. We've done an extraordinary amount with that pre-seed. We've gotten proof of concept for our first asset. We had a pre-IND meeting with regards to that first asset. We have done our digital and machine learning proof of concept. So we've gone through that cycle a number of times. And I think we're probably one of the first companies that I'm aware of to take machine learning insights about host range expansion, build those insights, and validate that machine learning is a great approach to actually help us to overcome one of the key challenges in the space. And we've generated revenue showing, proof of concept with regards to product market fit. So the current money that we're raising will allow us to do the full preclinical package and sort of on the first asset and proof of concept on the second asset. increase our data acquisition. So this is one of the key bottlenecks from a machine learning perspective, is we've got to really increase the amount of data that we're pulling in. And we've managed to shrink our data collection assay to about a square centimeter. And we can get thousands of assays in that footprint. And so we're wanting to expand that platform and collect more data, and then reduce the cycle time. So right now, sort of we're looking at 60 days from sort of printed to live virus, and we think we can get that down to 10, and then reduce it further down the road. We would like to increase the revenue on our platform and start to do more out licensing deals, and that will then, in about 16 months or so, lead us into our Series A, which we've got sort of a number of aspirations, which I won't go into the details just yet. If you are interested and you'd like to learn more, please reach out. This is my email here. We are also on Twitter and on LinkedIn. I think it's important to note that nothing that we've achieved happened by ourselves. It's only through the help and support of our funders, our partners, and collaborators that we've been able to do as much as we've been able to do. We're building a future that we're all excited about. And these are the folks that have helped us get to this point in time. And with that, I'm going to say thank you and wrap.
Thank you, Robert. That was a fantastic presentation. And it's so great to see Phage Research and CRISPR come all the way from, Delbruck, who started in California, all the way to finding new therapies for new anti-infectives and strategies for AMR. What challenges have you faced when you first decided to, form the startup? What have you been through? What advice do you have for young founders?
So I think the biggest challenge, there's two key challenges that we continuously struggle with. And the first is we're trying to develop a new antibiotic, and antibiotics are not very sexy. They traditionally haven't had a great ROI for a lot of investors. And so really, making the argument that if you pick the right indication, there is a good ROI. And there is, the traditional sort of poster children that get held up in the antibiotic space for why people shouldn't invest. Those are individual examples, and there are other approaches. So that's probably one of our biggest challenges, is getting people excited not only about the benefit to humanity, but about the potential ROI in the space. And I would say one of the second biggest challenges we've had is we're dealing with the technology around which there is understandably quite a lot of healthy skepticism, right? So phage have been around for 100 years, right? And I think there's been a number of waves of interest in the space, communicating to people that the key reasons that phage has not caught on in the past are technical reasons. And we now have the ability and the technical infrastructure to overcome those challenges has been a big challenge too. And of course, this is a hypothesis that we have. And we're working on de-risking it and evaluating it and testing it in the best way that we can with the resources that we have. But those are the two biggest challenges I would say that we face so far. But we're confident that we're going to give this a good shot, and we're hopefully going to develop a tool that we can add to our toolbox so that as we grow older, we don't have to worry about going back in time to pre-1930s when antibiotics weren't around.
Yeah, and you have assembled a great all-star scientific advisory board. You've got the backing of some of the big accelerators in the country, so it's fantastic. Really exciting to hear about your research.
Thank you. Much appreciated.
Thank you. And hope to stay in touch. Hope you're part of the MassBio community. So we'll be seeing you.
Great.
Thank you. And with that, we come to the conclusion of our second day of Innovation Week. We hope you'll join us again tomorrow for cardiovascular and metabolic disease and rare disease. Looking forward to seeing all of you back online tomorrow. Thank you, everyone.